Hey Tigers, how's it going? I hope you're doing well. I know this is an uncomfortable, awkward, and very confusing way to transition your grade 12 year. But it's a situation that we're all in, so let's do our best. So what my plan is for now, and this is going to keep changing as we go, no doubt, but we'll see how this goes for the first little chunk here, is I'm going to do these screencasts and I'm just going to record the lecture with my face down in the bottom right hand corner. I can't see you, and by the time you watch this, I've already long filmed this, but I'm going to sort of try to teach you the way I would have if you were right in front of me. And instead of filling out booklets together, I'm going to have a PowerPoint in front of you. So I will post all of this onto our Google Classroom. So this is going to be the PowerPoints that I'm going to sort of teach us from, and I'll just keep doing that for the remainder of however long we're out of class for. And so it shouldn't be a huge difference or change for you. And again, I'll give you a copy of these files on our Google Classroom so that you can download and kind of follow along as we go or make your own study notes. I'll also, as we do this, kind of show you videos on this screencast. Some I'll show in the actual screencast itself, and others I'll link to the bottom of the YouTube video, which I will post my screencast to, and then you can kind of click them as you see fit. I would highly recommend accessing every resource that I push out to you. So consider videos like almost mandatory as you will at this point. Even if I don't force you to watch it in this screencast, if I link it below or post it to Google Classroom, watch it. If I pause on a sample question, like over here, I might say, okay, pause the video here and do question number 2. Please do that. Find the time to do that. And email me if you have questions, as always. If you don't have my email, just type it into our EPSB Google Suites, or I'll obviously link it to our Google Classroom. But yeah, so that's how it's going to work for now. I'm going to do it all digitally. We'll see how it goes. I'll get better at as I go too, so please don't horribly judge how this goes the first time around. I will constantly reflect on how these looked and how well they were, and we'll go from there. I am trying to use this screencast program, and from my understanding, it's going to hopefully, for the most part, give me a closed captioning, like subtitles at the bottom once I'm done recording. And so you'll have a closed captioning of what I'm saying while I'm saying it as well, in case you do need to like mute it for whatever reason while you're listening. Again, I'm going to record this video of our lectures, post it to YouTube, and then I'll link you the YouTube link into our Google Classroom. And so my goal is that it'll have topic 3, here's the YouTube video lesson, here's extra lesson videos you could watch, here's some practice questions for you to do perhaps, and then hopefully, well, we'll just make it work. Obviously, this is going to take some personal oomph and desire on your part to keep going, but we'll figure it out together, okay? So with that being said, let's get going. So when we last spoke together in a regular classroom setting, we were here on topic 3, multiple alleles. So we're talking dihybrid crosses versus monohybrid crosses. So a dihybrid cross is something that's looking at 2 traits. So you've got 2 traits in front of you, and we're looking at, for example, what are the chances of having a kid and that kid's got green eyes and they have brown hair, right? Or they have a long nose versus a short nose and fat feet or skinny feet, right? 2 totally unrelated traits that we're looking at together. And so if I take a look here, as an example, let's say our first dihybrid cross we're going to look at, let's go back to Mendel's peas. He's got a thing for peas, and he's looking at peas, and he's like, okay, I got tall peas and short peas. So tall plants and short plants. And we're going to represent that with the letter T. Capital T, tall plant, lowercase t, short plant. That's 1 gene on 1 chromosome. On a totally separate chromosome is a totally separate gene, which codes for a totally separate trait, and that's the color of the pea plant. Yellow, big Y, or green, little y. So 2 totally different traits. Height, tall versus short, represented by T, and then color, yellow versus green, represented by Y. And now we'll cross them. So we've got a little bit of a more complicated looking cross here. We're crossing a tall yellow pea plant with a short green pea plant. Now you'll notice here, obviously, color coded the PowerPoints for you. If this is like a diploma question or you're working through practice questions, use your highlighter. Like use a color to separate your traits if you want, just so you can see the two traits separately. Really good skill to have as a visual person, as you know. I'm all about the color coding. Color away. So we're trying to do this cross. Tall yellow. So to be tall is homozygous dominant for the T gene. And then to be yellow, you have to be homozygous big Y for the yellow colored gene, a totally separate trait. So there's one individual there. And then we've got the other individual who is short and green. So little t, homozygous recessive for the short trait, and then little y, homozygous recessive for the green color. And so if we take a look here, again, just the same as usual, what we did in class together. Step one, make your legend. In this case, the legend was already given to us here. Big T was tall, little t was short, big Y was yellow, little y was green. That's our legend. Keep track of that as you go. Step two, you got to figure out what your genes are going to be that you're crossing in your Punnett square. Now, this isn't like a 2x2 square anymore like we were doing before with just the monohybrid cross, like, you know, 2 alleles on the top crossed with 2 alleles on the left. Now we're using 4 different alleles. So we have to find a new way of figuring out what the gametes are going to be. Like, what are you actually crossing in your Punnett square? What's going on the top and what's going on the left? And so we're going to use a method called FOIL. So that's what this is over here on the right. First, outer, inner, last. And it's just, that's the pattern. That's the pattern that we're going to actually do our gamete creating in. So just bear with me for a second, and I'll show you what this is going to look like. So it's going to get a little confusing before it gets easy, but take a piece of paper out. So like beside me here, I've got a notepad, pen, grab one. Grab a notepad, grab a pen, and try doing these things. Even if I don't tell you to pause the video, pause it and figure out, if you need to stop for a second, you figure out what you need to stop with, and then keep pressing play afterwards. So if we look at what's up here, remember that chromosomes line up totally randomly. Like, it's called independent assortment or random assortment. So when we line up everything on the metaphase plate during meiosis, and then in anaphase we split them apart, chromosomes are totally segregating and separating randomly. And so you could have any random combination, right, of those 4 different gametes. And so if we look at what foiling would do to create those gametes in terms of answering a question, this is a good, easy example to start. So let's say I've got a plant, tall, big T, and yellow, big Y. And I'm trying to figure out what the gametes are going to be in the square. So this pea plant is going to make a big T, big Y, a big T, big Y, a big T, big Y, and a big T, big Y. How did we get those? Well, we foiled it. So first, outer, inner, last. So first, we take the big T, and we're going to the first of the other trait. So there's your F, first. Your second, your outer, is going to be that same first T there, going to the outermost second allele, the big Y. So I had my F, right? So my first, so big T, big Y, that's that gamete right there. The O in FOIL, the outer, is going to be the big T going to the big Y. There's that big T, big Y right there. Inner is going to be the inner 2, so the second trait of the first, second allele of the first trait crossed with the big Y of like the first allele of the second trait. So there's your big T, big Y. There's your inner 2. That's that one there. And then last. So this second allele here crossed with the second allele of the second trait there, which happens to, again, be a big T, big Y, which is that last guy there. So first, outer, inner 2, and then your last. It's a pattern. You'll get good at it. This one's hard to see because they're all capital B, capital T's, capital Y's. Let's try another one. This one's probably going to make a little bit more sense. First, so big T, big Y. First, the first of each, the first allele of trait number 1 crossed with the first allele of trait number 2. Big T, big Y. Boom, there he is. Big T, big Y over here. That's your outer. So we did the first, the middle two, sorry, the big T, the big Y over here. Now we're going to do our outer, so big T, big Y. That's this one right here. Inner is your second allele of the first trait versus your, oh, my Siri is going off here. I got to turn that off. Inner, so your little T crossed with your big Y. That's that allele right there. It's a gamete, really. And then your little T, big Y right there. So foiled it. First, outer, inner, last. One more time. Let's try another one. Pause at any time if you need to, okay? So this guy here is going to be first, outer. So big T, big Y is your first. Big T, little y is your outer. Little t, big Y is your inner. And then little t, little y is your last, right? The last one. The last allele of trait number 1 crossed with the last allele of trait number 2. So right here, these four, big T, big Y, big T, little y, little t, big Y, little t, little y, those are 4 gametes that one pea plant, this heterozygous tall, heterozygous yellow pea plant, is going to produce. So let's cross it. What happens if you took this giant big T, little y, big T, little y pea plant, crossed it with another heterozygous for both trait cross as well? Here, you know what? I'm going to actually just go slideshow, view show. Let's not overwhelm you here. Let's just break this down into smaller chunks. There. Okay, let's start here. So I'm crossing a heterozygous for both trait individual with another heterozygous for both trait individual. So I got to figure out my gametes, foil it. Nothing crazy. First, outer, inner, last for this top guy here, this first one, this first pea plant, and then you're going to foil the second one, the second pea plant later. So we're doing the first pea plant first. It's going to fill in the top row. Doesn't really matter. Pea plants don't have genders. So oh, that's not good. Okay, so first, outer, inner, last. I've got my 4 gametes. I just foiled pea plant numero uno up there. Okay, I'm going to foil my second pea plant. Same thing. You're going to notice that you've got the same 4 gametes because we crossed the same heterozygous for both traits individual pea plant here with another of the same genotype, heterozygous for both traits. Heterozygote tall, heterozygote yellow. So they're going to have the same gametes because they're going to foil the same. Okay, fill in this Punnett square like you would have any Punnett square, even the ones you did in grade 9. Doesn't matter. Same thing. Just remember, write your capital letter first and your lowercase letter second, and keep your letters together. So you're going to write your T's together and your Y's together. So this first one, T down, T in. Y down, Y in. Boom. Okay, next row. T down, T in, big Y in, little y down, right? Right there. Cross this and this. Same thing, the whole row. I can do the whole thing. Same thing. Top down, left in. Top down, left in. Top down, left in. For both traits, key here, write your dominant trait first, and then write your, then keep your alleles together. So the T's stay together, big and then small, dominant and recessive, and then your Y's stay together, dominant, recessive. Do your whole square. So there's your square. It's overwhelmingly large, not overwhelmingly difficult. In fact, this is the hardest dihybrid cross Punnett square you will ever be asked to do. Is a heterozygote for both trait individual crossed with a heterozygote for both trait individual, right? You got heterozygous big T, big Y, little t, little y, crossed with another pea plant, which is the exact same, heterozygous for both traits. That's the most complicated Punnett square you're going to get, and even these ones follow a pattern, and that pattern is 9 of them are going to have one phenotype, 3 of them are going to have a different phenotype, another 3 are going to have a different phenotype, and then only one of them is going to have a very last recessive phenotype. Let me show you what I mean. Okay, so I've got 9, okay, I've got 16 total possible offspring here, right? When this tall yellow pea plant crosses with this tall yellow pea plant, I'm going to get 16 possible offspring. 9 of those 16 offspring are going to be tall and yellow. They're going to have at least one big T and at least one big Y. And then 3 of those 16 are going to be tall. They're going to have the dominant first trait, but the recessive second trait. So only little Y's. They're going to have at least one big T, but they're going to have 2 big Y's. So 3 of those are going to be the tall trait and yet have the recessive second trait, in this case, the color green. Another 3 of your 16 are going to have the opposite phenotype. They're going to have the recessive height trait, so they're going to be short, but they're also going to have the dominant second trait, so they're going to be the color yellow. So the 3 out of 16 that I colored in red here versus the 3 out of 16 that I've colored in green here, the difference between those two is that they flipped their phenotype. 3 of them are homozygous for the one trait, but dominant for the one trait, tall, but recessive for the second trait, color, in this case green. And then the other 3 are recessive for the first trait, so they have nothing but little t's in them, but they have a mixture of a big Y and or another big Y for their yellow trait. So they're going to show the dominant yellow trait. So that's like this one here, right? There's one of your 3. Recessive for the height allele, so he's short no matter what, but he's still yellow, big Y, little y. This one here, little t, recessive, so he's short for the first trait, but he's still yellow with the color, just happens to be homodominant for yellow in this case. And then there's your third. Still too little t, so still recessive short, but then you've got your dominant Y color there. It's heterozygous, doesn't matter. It's going to show phenotypically as short and yellow. Short and yellow, short and yellow, right? Your 3 out of 16 tall, you can find them in there too. You're looking for 2 with your little y. So big T, little y, there's one. This one over here, big T, little y, right? There's another one, tall and still recessive for the second trait, so green. And then your third one is going to be this one right down here, right? You've got your still recessive for the color trait, so green no matter what, but it's got a big T. Boom, there's your third. So there's your 3 that are tall and green, 3 that are short and yellow. Remember, 9 of them are tall and yellow, so 9 of them have the dominant trait for both no matter what. That's the majority of them, right? Anybody that's got a big T and a big Y. 2 of each, so homozygous dominant and homozygous dominant for both traits, or heterozygous for both traits, doesn't matter, as long as they fit the bill to be dominant for trait number 1, so tall, and dominant for trait number 2, so yellow, they're going to be part of those 9. The very last one here, there's only one of them. This is the easiest one to figure out. Recessive for both. So little t, trait number 1. Little y, trait number 2. They are going to be short, and they're going to be green in this case. So we got this phenotypic ratio, 9 to 3 to 3 to 1. You can memorize that. You can take that to the bank, that every time you're going to have a heterozygous crossed with a heterozygous, right? Heterozygous for both traits, crossed with a heterozygous for both traits, you're always going to get this spread. 9, 3, 3, 1 for your phenotypic ratio. 9 that are dominant for both traits, 3 that are dominant for the first trait but recessive for the second trait, a different 3 that are recessive for the first trait but dominant for the second trait, so opposite that first one, and then only one, always only one out of the 16 that's going to be both recessive in both traits, so short and green in this case. You can add them up, right? 9 plus 3 plus 3 plus 1, that's your 16, boom, done. Okay, example number 1. Predict the phenotypic outcome of a heterozygous, tall, homozygous yellow pea plant crossed with a, it doesn't say it here, but homozygous dominant for both traits. So big T, little y. So I'm going to look here. I've got my legend, step number 1. Step number 2, make your cross. So homozygous for the tall trait, heterozygous for the tall trait, so big T, little t, and then homozygous yellow for the color trait, crossed with a big T, big Y. There you go. Got to make your gamete, so foil it. First, outer, inner, last. So first, so I'm going to go big T, big Y. There's my first. Outer, right? Big T, big Y again. These ones, these outside ones. Inner, your little t, your large Y, right there. Those inner two. And then your last, your little t with your big Y. Little t, big Y. There's your last one there. Okay, same thing for the other one, but let's save a step. You don't need to do 4 gametes here, because look, these 2 big T, big Ys are exactly the same, so you don't need to include them because it won't change your probability in your gametes. So you can shorten your work a little bit. We've got 2 big T, big Ys, cut that extra one out. You've got 2 little t, big Ys, cut the second one out. Your square is automatically smaller now. And I'll show you what that looks like in a second. Let's look at this guy. Your second cross, your big T, big Y, big Y, your tall and yellow homozygous for both. Foil it, big T, big Y, big T, big Y, big T, big Y, big T, big Y. Foiled it, found your gametes. All 4 of those gametes are the same. Cross all 3 of them out. That one's crossed out, that one's crossed out, that one's crossed out. You only need one of them right there. You only need the one. So really, this Punnett square is actually going to be almost smaller than the ones you did in grade 9, believe it or not. Boom. There, see, you got your gametes from your first guy right here. So big T, big Y, little t, big Y, right? Those are those 2 gametes we just foiled out. We didn't have to include the doubles of them because it's redundant. And then look at that. Look how easy that is. Big T, big Y right there. Because all the other 3 for that second P plant were the same. So my Punnett square just became easier than something you've even done in grade 9. So dihybrid crosses, looking at 2 traits, don't have to be hard. The hard ones, like you already saw the hard one, the 9-3-3-1 spread one, where you're crossing a heterozygous for both traits with another heterozygote for both traits. That's the hardest and the biggest, and even that has that pattern in the phenotype, right? The 9 to 3 to 3 to 1. But in this case, boom, you just got an itty bitty little Punnett square, same as usual, top down, left in. There's one possible offspring, and boom, there's your second possible offspring. So one offspring is homozygous tall and homozygous yellow. The other offspring is going to be heterozygous but still tall and homozygous yellow. So all of them are tall, all of them are yellow, but your genotypic ratio is 1 to 1. Okay. Okay, a couple more examples. Example 2, a dwarf red snapdragon is crossed with a homozygous tall white plant. Okay, homozygous tall and also white plant. Determine the phenotype and genotype of the F1s, of the first generation, fleal 1. So tall is dominant, red is incompletely dominant to white. Okay, so incompletely dominant. They're going to blend. If you have red and white together, incomplete dominant says they blend. You're going to probably get a pink somewhere up in there. There you go. So step 1, always write your legend. Tall, big T, dominant, little t, we'll call it short or dwarf in this case. And then remember, we did a couple examples of these in class before we kind of broke for this COVID situation. But I've got CR, and that's a red plant, homozygous for both. If you're homozygous Ws, then you're going to be a white plant. But if you got one of each, because it's incomplete dominance, and they told that to us in the question, it's a pink plant. Step 2, do your cross, okay? So they said to me, this looks complicated, but it's not, right? Just go back to your legend. They said it was a homozygous tall plant. Crossed with a dwarf red snapdragon. So our dwarf red snapdragon is, well, dwarf, you have to be little t, so there's what that comes from, little t. And then CR, well, it's red, so there's only one genotype for a red snapdragon, according to this legend. Same thing for your homozygous tall white plant that they told us, right? Homozygous tall, big T, has to be, because they said homozygous, and then it's white. Okay, foil it as per usual. Don't get confused with the lowercase letters and the superscripts, and like, don't sweat it. Just business as usual, but mind your letters here, like be detail oriented. First, outside, inside, last. Foil it. Again, make your life easy. Don't do a giant square if you don't have to. All 4 of these gametes are the same. They're redundant. Cross 3 of them out. You don't need them. We're just looking at that one. Little t, CR. Foil your second one. First, outer, inner, last. Again, all the same. You thought the last example was easy? Check this example out. All 3 just got canceled out. We don't have to write them. You could totally do another giant 4x4 Punnett square if you wanted, but I mean, they'd all be the same. So why not just make your life easy? All these gametes are the same, get rid of all but one of them. All these gametes are the same, get rid of all but one of them, and then do your cross. Boom, look at that, 1 by 1. Now again, are you wrong to do all 4 TCRs and all 4 TCWs, like going down the left and over the top, and not canceling this out? No, you're not wrong, but you're just wasting time, because you're going to get the same answer no matter what. All of them are going to have the same genotype. If you don't believe me, try it. They're all the same. So top down, left in, boom. There's your guy. He's heterozygous, but he's still tall. And he's heterozygous for the color, one red allele, one white allele. They already told us, hey, these are incompletely dominant. It's a pink flower. So there's your phenotype, genotype right here. Done. Example 3. So I've got a crested head is produced by a dominant allele, and a plain head is produced by a recessive allele. So we're looking at like chicken head shape, basically. Black feathers are dominant to red feathers. So we're setting up some legend stuff for us here, which is good. And so we have a homozygous, red-feathered, heterozygous, crested head bird. Okay, that's a lot of words, but very helpful. Okay, homozygous red with a heterozygous crested-headed shape. So I already know both traits and the genotype for both. Is mated to a heterozygous black, plain-headed bird. And they want the phenotype and the genotype of the F1, so the first generation. Okay, so do your cross, do your legend. Foil it for the first one. Again, any doubles you have, it's redundant. Make your life easy. I had 2 little b big H's, cross one out. I had 2 little b little h's, cross one out. Do your foiling for the second one. Figure out that meet arrangement. Same idea, right? 2 big B little h's, cross one out. 2 little b little h's, cross one out. Boom. Classic looking square now, right? Draw one alleles on the top. Draw the other allele on the left, on the left, and then fill out your Punnett square as usual. Write your dominant letter first, keep your traits together. So big B, little b. Big H down, little h in. Top down, left in. Top down, left in. Top down, left in. The question's asking, hey, what are the phenotypic and genotypic ratios? So boom, here we go. Phenotypic ratio, I've got, one black crested head, because he's got a big B and a big H, and H was for the head shape, so he's crested. I've got one black plain head, this one over here. I've got one red crested, so red and crested, and I've got one red and plain, one homozygous for both traits. Not too bad, right? Genotypic ratio, I've got one of each, they're all different. Okay, example number 4, a blood typing one. Great questions, right? We know that we got to be mindful of our letters or superscripts for blood typing questions. So draw yourself a legend. We've got a rhesus as well. They gave us a little bit of a legend here for the rhesus. Rhesus positive is dominant to rhesus negative. And this question is saying, hey, you're crossing a woman who's type AB negative blood, and she's marrying a man who is heterozygous for blood type A. So she's heterozygous for blood type A, but is homozygous for the rhesus positive factor. Determine the phenotypic and genotypic ratios of their F1 or first generation offspring. Business as usual. You wrote your legend, you wrote your cross, you got to foil it. So go ahead and foil it. Any redundant gametes, get rid of them. So boom, boom, gone. Why make more work for my cross if I don't have to? Same thing here. Eliminate your redundants. Got a simple 2x2 Punnett square now. So there's we like to write the woman on the top here. So there's my woman blood type for both her gamete options. The man's on the left hand side there. Business as usual, guys. Top down, left in. Keep your traits together. We've got one that is clearly homozygous for blood type A and rhesus positive. Heterozygous for rhesus positive. Another AB rhesus positive. We've got a type A rhesus positive, and we've got a type B rhesus positive. So type B rhesus positive, a type AB rhesus positive, and a type A, 2 type A rhesus positives. Because we've got this one here and this one here. So there's your 2 type A's, your 1 type B, AB, and your 1 type B. But since all the actual genotypes are different, it's a 1 to 1 to 1 to 1. So you have 2 that phenotypically are rhesus positive type A, but their phenotype is actually, although the phenotype is the same, their genotype is different. So keep that in mind. Done. Just a little example here. I mean, you can pause it here and take a look, but this is just showing exactly what we've done. So a summary here. Most complex Punnett square you could ever do is a 2-parent heterozygous for both traits cross, right? So that's where you're going to get your 4x4, right? Two heterozygous for both trait, crossed together. 4 possible gametes from each parent. When you go to foil this guy and you go to foil this guy, you're going to get 4 gametes, and they're all different. And these 4 gametes, all different. You cannot eliminate if it's heterozygous for both traits, which is why the square is so big. It's a 4 by 4. But don't forget that even then, there's rhyme and reason. You're going to follow that 9, 3, 3, 1 phenotypic ratio. Right? So this ratio right here. 9 are going to have the dominant trait for both. 3 are going to have 1 dominant, 1 recessive. A different 3 are going to have the opposite dominant and the opposite recessive. And then only one of those 16 is going to be recessive for both traits. Doesn't matter if you're looking at peas or humans or dogs or whatever. If they're heterozygous for both traits, crossed with a heterozygote for both traits, you're going to get this 4 by 4 Punnett square. 16 possible options, but they follow this 9 by 3 by 3 by 1 phenotypic ratio. So here's an example here. This is not a case where you have a heterozygous for both. So you're going to have fewer possible gametes. So the Punnett square is automatically smaller. And don't forget, eliminate the redundant duplicate gametes. Right? So there's your cross for that one. So hopefully that's a little clearer here. There is a Bozeman science video on YouTube as well. I'll try to remember to link this down to the bottom of my YouTube video for you if you want more practice with it. And he's got some great videos, obviously, where he works through them. So give us a shot. See what you can do. We'll talk about lethal alleles, I think, in another video here. I'm just going to cut the video here. I do want to just direct you to a dihybrid and 2-trait amoeba sisters video here. I will link this in the description, but if you just Google or YouTube search dihybrid and 2-trait crosses amoeba sisters, great video, 8 minutes, worth your time. You guys should have a copy of this book printed still, but if you don't, or if it was at school or whatever, I have posted this onto Google Classroom, so just go ahead and search for the Mendellian genetics booklet. There's no page numbers on this booklet, unfortunately, but there's lots of questions that you can do. All of these are possible here. Question 4 is a really nice one. Question 5, question 6. You're more than welcome to pause this video too at any of these questions and take a peek and try them yourself. And I'll just keep scrolling down. You have access to this. The lethal allele ones I'll get to in the second video, but there are answers at the end of the page there, and I'll just kind of leave that up on the screen so you can hit pause if you need the answers for any of the ones you saw. But I hope this was helpful. This is just video number 1, you guys, so bear with me. I'm learning how to do these videos too. If you have any recommendations, let me know. Great. 30 minutes is a good time, I think, to cut it at. I'll try keeping them around this time because you'll get bored if you aren't already. But I love you. Thank you. And we'll figure this out as we go. Okay, guys?